We've taken you inside Anatome to introduce you to your protein self, to help you understand how the next generation medicines of biotechnology will affect you and our society. With your better understanding, society will be able to contain the risks of biotechnology so that we can all safely benefit from its vast rewards. These new medicines will affect your protein citizens, the active molecules that create everything about you. Composed of folded amino acid chains joined together to form complexes, protein citizens use energy to perform work. They read RNA instructions to build more of themselves. They read DNA instructions to build RNA. Protein citizens cannot exist without DNA and RNA, so let's broaden our view to help you understand yourself in terms of all three. We'll introduce you to the amazing relationship that protein, DNA, and RNA share, and show you how everything about you has come to depend upon that relationship, including your health. But first, we have one loose end to take care of, for we have yet to explain how DNA comes into existence. Is there another molecule performing this task? Watch how Cell City reacts after powerful sunlight breaks through its roof and damages its DNA. Protein citizens quickly arrive to repair it. They read the undamaged DNA strand to determine which nucleotides they must link together to replace the damaged DNA strand. Only complementary nucleotides will work, of course, so their job is made easy. Now take a look at this cell. It must create a copy of itself to replace a missing neighbor. It unpackages, uncoils, and divides its DNA, then uses each strand as a template upon which to construct a new complementary mate. Once again, protein citizens accomplish these tasks. DNA polymerase links together complementary nucleotides, one after another, to create a complementary strand for the top strand of DNA, and then the bottom. The end products are two double-stranded DNAs, each an exact copy of the original. Other proteins package these into chromosomes. Soon after, protein citizens create copies of everything else in their city, which they divide into two identical cells, one of which fills in for the missing cell. So rejoice! Protein citizens create everything about you, including your DNA. With no new molecule to discover, we can now explore the very core of your being, where protein, DNA, and RNA share a fascinating relationship. We know that protein reads DNA to make RNA, and that protein reads RNA to make more protein. That protein, as we have just explained, creates DNA. And with more DNA created, more RNA can be created. And from that, more protein, DNA, RNA, so on and so forth. Well, isn't that amazing? Working together, protein, DNA, and RNA can create copies of themselves. In a word, they can replicate. If we submerge them in salt water, enriched with various factors, including nucleotide and amino acid building blocks, and an energy source, they will replicate continually. This replication is occurring inside of you right now, and inside of every other body that scientists consider living. That being the case, scientists believe that the first life form on Earth was probably a simple association of these three molecules replicating. Let's imagine that association looked like this, and call it a geneSpring. Somehow then, according to scientific theory, as the geneSpring replicated through time, it changed to become human. But how could such a simple molecular association ever become something so complex as a human being? Let's travel back in time to see how this might have happened. Billions of years ago, much of the Earth was submerged in molecular soup, full of simple molecular building blocks whose special chemistry allowed them to assemble into chains and then create copies of those chains from other blocks around them. Eventually, they became organized into the first geneSpring on Earth. Here it is, composed of primitive DNA, RNA, and protein, replicating. And here comes Mother Nature to welcome it. Though the geneSpring can't protect itself from nature, it is not easily wiped out, for its offspring continue to replicate throughout their liquid world, making them difficult to kill all at once. Still, nature strikes and takes her toll, but misses once in a while, damaging rather than destroying a geneSpring. True to its chemistry, that geneSpring continues to replicate. It begins another round, first dividing its complementary double-stranded DNA into 2 strands, then creating a complementary mate for each. So far so good. One perfect copy of the original DNA has been created. But problems arise when trying to create the second. The damaged nucleotide on the second template trips up the primitive geneSpring polymerase, causing it to reread part of the strand and mistakenly insert additional nucleotides. Then, it confuses the polymerase into selecting an uncomplementary match for it, a cytidine rather than a thymidine. When finished, the geneSpring protein has created a second DNA that is not an exact copy of the original. But that doesn't stop other geneSpring proteins from reading its DNAs and replicating new RNAs from them. Or from translating those new RNAs into new proteins. As this round of replication completes, 2 new genesprings arise. One, a perfect copy of its parent, the other, completely changed. Not only because of the insertion of 6 additional DNA nucleotides here, and a mismatched nucleotide within this group, but because these DNA changes lead to RNA changes as well. For RNA is replicated from DNA, nucleotide for nucleotide, and must therefore reflect DNA's changes, more or less, which appear here as 3 new codons. Of course, the new geneSpring protein arising from this RNA must also reflect those changes, for the new changed RNA directs its replication, and additional codons in RNA must translate into additional amino acids. Thus, we see a new protein take the place of the original, containing 2 new additional amino acids inserted within this arm and a changed amino acid in this arm. We've just passed an important step on the geneSpring's path to become human. It has mutated. Nature has reshaped its DNA, and that has led to a change in its form, and brought a new protein into the world. If this new protein does not lessen the geneSpring's ability to replicate, then the geneSpring will simply follow its calling and begin spreading its mutated form throughout the world. But suppose a second natural challenge results in this. The addition of this new DNA, this RNA, and these amino acids to the geneSpring. Then mutation is not a good thing, for this geneSpring's protein will be so changed that it can no longer function. It cannot help its geneSpring to replicate, and that means game over for this geneSpring. A mutation might not seriously affect a geneSpring, or it might fatally cripple it, but it might also help it. See how these mutations have turned this geneSpring into a faster replicator? Now it can make more of itself more quickly and spread its line throughout the world more quickly. This makes this geneSpring more difficult to kill off and more likely than its weaker parent to survive and experience additional strengthening mutations. Scientists call this shaping-strengthening process evolution. Nature mutates the geneSpring's DNA. Those mutations give rise to new proteins. If those new proteins make the geneSpring stronger, it survives and continues to evolve. Its DNA and RNA stores grow. Its proteins continue to increase in size, number, and complexity, and its population spreads throughout the world. From this to this, our geneSpring is becoming ever more human, for it is now capable of making many more proteins that began as accidents of nature, but continue to exist and evolve because they help their geneSpring to survive. They are now protein citizens, capable of creating large structures out of other types of molecules that become layers of protection for their geneSpring and their city, like this nucleus and these cell walls. They further evolve into proteins that can divide one cell city into many and cause cell cities to group together and communicate with one another. As protein citizens further evolve, they change the shapes of their cell cities, which enable them to perform bigger feats. They can now sense their environment, coordinate group action, and flee from danger. Under nature's guiding hand, the geneSpring has evolved into a multicellular organism, able to function, survive, and further evolve because of the protein, DNA, and RNA still replicating at its core. Millions of years later, that multicellular organism has become you. Your body and mind have become so fit that you now have time to relax and consider how to tap their full potential. But to accomplish this, your body and mind must be operating in perfect health, which means, of course, the organ systems of which they are composed and the cells that run those organ systems. Through this presentation, we've tried to take your understanding even deeper and show you that all of the above can only operate in perfect health when their ultimate makers, your protein citizens, DNA, and RNA, are themselves operating in perfect health. Which brings us to biotechnology. The most effective treatment for any human disease or illness will specifically target only those proteins, DNAs, and RNAs involved in sickness and restore them to perfect health, without affecting anything else in our bodies and without subjecting us to the unnecessary and unwanted side effects of chemical sledgehammers. Medical scientists are rapidly developing this revolutionary biotechnology. We'll examine the progress they are making in our next presentation, Tweaking the geneSpring.